Thank you. Among the open questions in neuroscience, I believe the functional sleep remains perhaps one of the most embarrassing. We don't know why every night our brain needs to put us and presumably itself to sleep for several hours. And there is every indication that sleep performs an essential function if we only knew which one. The indications are that it is, first of all, dangerous. Obviously, you are available to predators if you fall asleep and you don't respond to stimuli. It is pervasive. We do this from the cradle to the grave. Every animal that has been studied carefully does so. Going from fruit flies and now several other species have been shown to sleep to ourselves. There's basically no single exception. It is also irresistible, as we all know. But if you try and do experiments to keep animals awake, or humans awake for that matter, there is no way that you can overcome the need for sleep. You can even use pain, shocks. At some point, the animal will fall asleep. It's also tightly regulated with a big portion of the brain, and certainly the brainstem and the hypothalamus. All kind of complicated connectivity is set up to put us to sleep and then wake ourselves up. And finally, if you don't sleep or you sleep too little, it's clear there are serious consequences. According to some people, death would be one consequence. In fatal familial insomnia, after a few months of essentially lack of sleep, you die. Rats die after 2 weeks. But the most obvious consequences are cognitive consequences. There are all kinds of problems. We become extremely bad cognitively. We make mistakes of all sorts, and we become extremely irritable. So now let me, before I go and discuss the issue of the functional sleep, remind you with some perhaps novel work of what's happening in the brain when we sleep. This is an indication of the fact that when you go from wake to non-REM sleep, which is 80% of sleep, in humans, most mammals and birds, what you see are those big slow waves in the EEG that are abundant early in the night and then less so later in the night. And corresponding to those slow waves, you actually see units, neurons. This has now been shown in humans and cats, rodents, monkeys. They fire roughly at the similar firing rate as in wake. They just have these brief periods of silence lasting 100, 200 milliseconds, something like that. And then they resume firing. And intracellularly, as shown by Mircea Steriade and his collaborators, what happens is neurons are firing tonically and then they stop. Briefly, they hyperpolarize 15, 20 millivolts, and then they go back. This is what's happening 1,000 times per night in our brain. And this is triggered and associated with changes in neuromodulators, like the ones that Michael just described, acetylcholine, norepinephrine, serotonin, histamine, orexin, and so on. They all change dramatically between wake and sleep, and obviously produce the very different way of functioning of the brain. Now here, I want to use an old large-scale simulation with many tens of thousands of neurons and millions of connections of two cortical areas, like a primary and a higher order visual area, and a piece of thalamus, the LGN and the reticulothalamic nucleus, divided into 3 layers. And just to give you a feeling of the tonic firing during wakefulness with the corresponding EEG, or local field potential. So it's the typical low voltage fast activity of wakefulness. And neurons fire in this random way, as if we were imaging them with optical recordings. And then we turn the neuromodulators down and we send the model to sleep. And when we send the model to sleep, it continues firing, but there are these very brief interruptions. It happens roughly once every second. And that is most of what sleep is made of. There are additional things like sleep spindles, etc., but the slow waves that correspond to neurons doing that is the major feature of sleep. Again, if we knew why the brain is doing that, because when it is doing that, it is also obliterating, to a large extent, our own consciousness. So now we know a few more things about this. One of the things we have learned is that each of these slow waves, especially the big ones, start in a different place in the brain and then travels. It's literally a traveling wave that invades other regions of the brain. And when doing source modeling, we actually saw that many of these slow waves, the big ones, start around the left insula, also on the right side, so it's distributed, but there are some hotspots. And then they travel and they propagate throughout the brain along the main superhighways of the connectome, if you wish, especially along the medial surface of the hemispheres, and then they spread out laterally, invading much of the brain. Now, a recent finding that was done in collaboration with Itzhak Fried by Yuval Nir is that if you record in humans, in, let's say, 10 different regions in the brain, when the human is entering non-REM sleep, what you find is that while there are indeed global waves that invade most regions, the ones we typically see with the high-density EEG on the scalp. There are plenty, in fact, the majority of waves that are local. They may happen in one brain region and not in others, or in two brain regions and not in the others. So a lot of what you see with the scalp EEG is actually a reflection only of the biggest phenomena. There are local waves happening all the time throughout the human brain, and as we now know, also the rat and the mouse brain. This is a recording again from Yuval Nir of a rat. A is auditory cortex and F is frontal cortex. You see the EEG, you see the local field potential, you see the multi-unit activity, and you see individual spikes. And you can see these periods of silence in the array recordings. Those are the downstate of the slow oscillation underlying the slow wave. And very often, those downstates happen in one region, auditory cortex, for instance, and not in another region, prefrontal cortex. So this is happening 1,000 times a night in different ways, in a sort of a synchronous manner sometimes, throughout the brain. Why? So the enigma of sleep function has been there for a long time. All kinds of ideas have been proposed. What I'm going to discuss today is the Synaptic Homeostasis Hypothesis, which has been an attempt to understand what may be the fundamental, the core function of sleep in every animal. And the idea here, in short, is that sleep is the price we pay for plasticity. Of course, plasticity should happen primarily during wake when we adapt to the external world. Now, I will not discuss the theoretical motivation for this, except for this very brief sketch, which suggests the following, that neurons either spike or they don't. That's how they communicate. Spikes are more expensive than non-spikes in terms of their postsynaptic consequences, in terms of energy and everything else. So spikes should be reserved, and as far as we can tell, they largely are, to signal important events, events that convey a lot of information. Now, when you need to adapt to a changing world, which is basically all the time, especially during development, what you need to do is to shift strength of synapses or even add synapses to make sure that you're firing for important events, for important changes dynamically in the environment. And you do that, and every neuron tries to do that to make sure it signals important stuff downstream in this complicated brain where every neuron is in a sea, immersed in a sea of other neurons. It doesn't know what it's getting, from where, it doesn't know what it's signaling, and doesn't know where it's sending it. So in all of this uncertainty, for any learning system which is as complicated as a brain, there is a problem in the end, which is you tend to strengthen to make sure you signal, but that becomes biologically untenable because stronger synapses consume more energy, occupy more space, require more supplies, and finally they saturate signal to noise. Basically, then neurons start firing for everything, and that can't be good. So there is a need for renormalization to make sure that total synaptic strength is constant. And we think that that renormalization is not only essential, but it better happen offline, when you actually can sample in an unbiased way the environment of a neuron. And every neuron by itself does it in the course of the night. That's what we think sleep is fundamentally for. And without, again, elaborating on the theory, let me just then illustrate in practice what this hypothesis suggests. It suggests that at the minimum, during wake, there is a net increase in synaptic strength, potentiation, and during sleep, this is renormalized to go back to a baseline value, which is sustainable biologically and from the point of view of information transfer in the brain. By synaptic strength, I refer both to the molecular aspects, like the number of AMPA receptors and their phosphorylation state, and to the electrophysiological aspect, of course, how effective is a presynaptic spike in producing postsynaptic depolarizations. And here, I want to very quickly go through evidence about this. Molecular evidence actually started a long time ago. You see there sections through a rat brain, and we see the expression of FOS in wake when it's white and in sleep when it's not white at all. Many other genes behave like that, and here I put a very nice recent study from the Allen Institute in which, in a much more thorough way, many different mRNAs have been mapped. And there too, ARC, for instance, is much more expressed in an animal who has been awake than in an animal who has been asleep. The general rule is, while many genes change, the one that seems to change most dramatically between wake and sleep are genes involved in synaptic potentiation and that kind of plasticity. Here you see, for instance, the phosphorylated Krebs in the cortex is high in wake and is very low in sleep. And similarly, BDNF and BDNF-marked fibers are much higher in wake than in sleep. Here you see a more detailed study using synaptoneurosomes of the density of AMPA receptors, GluR1, And they are much increased, like 50% or 80% in the waking cortex or hippocampus for that matter, compared to a sleeping cortex. And a more recent study by the group of Ulrich has also shown something like this with a completely different technique. I want to move briefly to the electrophysiological evidence, and I can tell you already it goes in the same direction. Here is a recent study in which we looked at minis, that is the spontaneous release of transmitter. You can see both the amplitude and the frequency of the spontaneous release is higher in slices. This is both mouse and rat of animals who had been awake in the previous 2-3 hours than animals who had been asleep. And here is the slope of the evoked potential, a classic measure of synaptic efficacy. So you give an electrical stimulation, you see how big and how steep the response is. And after an animal has been awake from W0 to W1, the response gets bigger and steeper. After an animal has been asleep from S0 to S1 for 3-4 hours, the response goes down. And in humans, you can see something very similar. You use, instead of electrical stimulation, you use TMS, and then you use high-density EEG to record the response. And again, the more you stay awake, the more excitability goes up. The response gets bigger and steeper, and then you sleep, and it goes back to a normal level. The most recent evidence that we have obtained about this is actually structural evidence. We reasoned that if these things change at the functional and molecular level, maybe also they change at the structural level. Some indication, this is burp, which is a very strong presynaptic marker in fruit flies. Some of the indications are that burp protein is now highly expressed, for instance, in all over the brain of a fly who has been awake, and much, much less in the brain of a fly who has been asleep. We showed quite some time ago that flies sleep very much like we do, so this is an indication that even at the level of synapses, things change in the same direction. And Dan Boucher and Chiara Cirelli recently were able to look at this using confocal microscopy, and here focus on one particular area of the fly brain, lobular plate, visual system neuron number 1, but it's true in 2 different structures that we analyzed. You can see the dendritic branching and the punctae in an animal who had been asleep, then an animal who has been exposed to a typical enriched waking environment. And you can see that the number of synapses goes up as well as the richness of the dendritic branching. When the animal goes to sleep afterwards, the branching and the synapses go down. That does not happen if the animal is kept awake, meaning sleep is actually necessary to produce the synaptic pruning and branching simplification. You see something similar now. This is another recent study that was done by Stephanie Marais and Ugo Faraguna using 2-photon imaging on the mouse up there, sort of like we saw before. We did several sessions in the same animal, that's pretty hard to do, and counted synapses in adolescent mice. And after they've been awake, the number of synapses tends to go up slightly. And after they've been asleep for, say, 7 to 8 hours, it tends to go down. So there, too, there is a change in the number of the synapses as a function of having been awake and having been asleep. And perhaps one of the most unexpected to me findings just came out by a Japanese group of Yokoyama and co-authors have shown that the number of neurons in the olfactory bulb, which is, as you know, always undergoing very, very strong turnover, is heavily regulated, so lots of neurons die. Well, it turns out that they die in the postprandial sleep period. So they die, they disappear, they are selected away when the animal sleeps. So this pruning, so to speak, seems to be rather universal in the fruit fly, in the rat, in the mouse, and it is also pretty extreme at times, including the number of neurons. So now I want to move briefly to a second aspect, a corollary of this hypothesis. If what changes between wake and sleep is the net synaptic strength and perhaps even structural markers of that, can we see that expressed in the usual parameters that have been used for many, many years to record sleep, that is the slow wave activity in EEG? Well, the hypothesis says that if you strengthen during wake, then this increased net synaptic strength is going to produce stronger coupling among neurons, which should, at the minimum, result in a higher synchronization and therefore in bigger and steeper slow waves when you go to sleep. Then the renormalization of synapses during sleep, so that they tend to go down, will reduce this coupling and reduce the amplitude of connections down to a reasonable level. And that is what we first tested with large-scale models. If we increase the strength of connections to plasticity during wake, when the model goes to sleep, as you saw before, the slow waves are bigger and more synchronous. And then at the end of sleep, they become smaller and less synchronous. And this fits with what we actually measure in the slow wave activity in both the model, rats, and humans. Now, if you try and manipulate this process, you can actually see some local component to it. You can, for instance, ask an animal to locomote a lot, which fits very well with what Michael was saying a moment ago. That would produce several marks of plasticity enriched in the cortex, like BDNF. And as a result of that, when the animal goes to sleep, the slow activity will be larger. And if instead you keep an animal awake for exactly the same duration, but it's doing very little, it's moving very little, then the BDNF will go up less, and so will other markers of plasticity, and the slow activity in the night will be reduced. We can actually, interestingly, I don't have the slide here, but lesion the noradrenergic system, one of these key neuromodulators, and that will produce similar results even in an animal who is locomoting. So it will decrease the amplitude of this. In a local sense, you can do a learning task. Here is a rotation learning task done by humans in front of a computer where they sort of adapt unconsciously to a rotating display. And the area of the brain that's involved in learning that task, which we sort of knew, is right parietal cortex. After you learn and you go to bed, shows bigger slow waves. Conversely, if you immobilize an arm, the corresponding contralateral somatosensory cortex will show less slow waves when you go to bed. So this indicates that the kind of plasticity and where the plasticity occurred during waking is simply read out by the amplitude and steepness of the slow waves during sleep. You can see this here, for instance. We apply this in behavioral therapy for aphasia. So aphasic patients underwent a very intense behavioral therapy in which they exercise and exercise. Then they go to bed, and we can see that the relevant areas show more slow activity. So presumably this could be a way clinically to test whether plasticity occurred, how much of it, and by the way, this predicts recovery of function. I want to finish by telling you only in a few words what are, therefore, the consequences of the net increase in synaptic strength that I believe happens in every animal when we learn, for the theoretical reasons I only briefly sketched. You end up in the evening with more and or stronger synapses. Those consume more energy, even doing nothing. They occupy more space, and we saw from Sinofsky's presentation this morning how little space there is there. They will require a lot of supplies, going from mitochondria to membranes to proteins that need to deliver to the synapses. And by constantly increasing, they will decrease the signal-to-noise ratio. So when sleep comes in, it renormalizes, we think in a very smart way, the overall synaptic strength, putting all of those functions back to where they need to be. I want to finish, instead of discussing so much the evidence for how sleep fulfills its functional role there, by asking what might happen if you don't sleep long enough, and what is a typical reaction of neurons. So this is a recent study by Vlad Vyazovsky, in which, you see that a rat who is not as awake as he looks. In other words, he looks completely awake. He runs around. He does all the normal things rats do. And his EEG is a perfect waking EEG. But if we keep him awake for 3-4 hours more than he normally would, what you can see is that after sleep deprivation, you start seeing off periods, very much similar to those that normally happen only during sleep in a completely awake animal. And this can be global, but more often they are local. They may happen in one brain region and not in another. So you have this rat running around in whom the behavior is awake behavior. The EEG is a wake EEG. You couldn't really tell that anything is wrong. And one particular area just goes offline briefly for a fraction of a second. When that happens, the rat makes mistakes if it happens in the area that's controlling behavior at that point. So it is not a long shot to imagine that something similar happens to us. And we now have indications that at least at the EEG level with high-density EEG, if, for instance, you stay awake too much, and in one case you do lots of talking and listening in dim light, and in the other case, instead, you do a lot of driving simulation with no talking and listening, you actually tire off, as expressed there by waking theta power, progressively more certain regions than others. Everything gets tired, but some regions more than others. And those sudden theta waves in the wake EEG probably corresponds to those off periods at the neuronal level, if we could ever show that also in humans. And when that happens, we make mistakes of all kinds. Thank you very much.